Hi, my name is Rachel, and I am a senior biochemistry major at Texas Tech University. As a Sizer SSO scholar, I research in a clinical microbiology lab under Dr. Ali Smith, and my research involves chronic wounds and biofilms. One of our main goals is to improve care and diagnostics of bacterial infections. In this video, I will be giving a short tour of our lab. We have a lot of stuff that is essential to our research, and I will be going more into depth about some of the stuff that we have. If there are some things that I do not show that you see in this video, it might be in Isaiah's video, who is also another Sizer scholar from this lab. So go check out his video too, and if you have any questions, you can contact us or Dr. Smith, and we would be happy to answer them. The first stop in this lab is at the walk-in incubator. This incubator is really old, but it has a lot of space to store anything that we need to grow. The machines on the table are called shakers, and we use them to help some bacteria aerate and grow better. Moving on, we come to our autoclave. This machine uses high pressure and temperature to sterilize our glassware so we can use them in our experiments without worrying about contamination. Right next to our autoclave is our magic water or double distilled water. We use this water to fill the autoclave with and for mixing broths and augers, or even for making our 70% ethanol so we can sterilize our hands and wherever we'll be working. Next to our magic water is a fume hood. We use this one sometimes, but we also go to the lab next to ours and use their UV fume hood to also sterilize certain things. Across from our fume hood is our deep freezer. This freezer has to be kept at around -80 degrees Celsius. We mainly store cryos of different bacteria species, and when we need a certain species, we take out the cryo to make an overnight culture out of them. To me, this is interesting because when the cryo thaws, the bacteria can resume growth again. Now we are going to come out of this room to where our main shaker is. This shaker is also an incubator, and as you can see, we are growing biofilms in there right now. Our lab is sponsored by Moleculite, which is a company that creates clinical imaging devices for determining if wounds are infected or not. I'm going to show how the camera works briefly through a Staphylococcus aureus streak plate that is plated on two different augers. On the left, the plate is a blood auger, and on the right, the plate is a porphyrin test auger. When we turn off the lights, we can see that the bacteria on the porphyrin test auger fluoresces red, but the bacteria on the blood auger does not. This is because the bacteria on the left were induced by nutrients in the auger to produce porphyrin, a component that is naturally produced in our bodies through heme synthesis. Porphyrin is excited by a violet light, which is why the light coming from the camera looks violet, and then the emission light coming from the bacteria is red. Along with Moleculite, we have several projects in our lab, so we never really run out of things to do. This lab is hands-on and requires a lot of time commitment, but what we do can truly be meaningful one day, and that's why research is really exciting. If you are interested in joining this lab or for more information, this is Dr. Smith's contact information and my own. I would also like to take this time to thank Sizer, Moleculite, Dr. Smith, and the TTU HSC for giving me the opportunity to create this video, as well as to be able to conduct research as an undergraduate student.